All right, so this video we're going to be talking about the Calvin cycle. So let's kind of take a second and just revisit what is the bigger picture of the Calvin cycle. Now, previous video talked about the light reaction in making ATP and NADPH. Oxygen is a byproduct of this, but the whole point for this step is you make NADPH and ATP to power the Calvin cycle. Okay, so one common question really is why are ATP and NADPH needed? So one thing to consider is, well, you're going to be taking in some carbon dioxide and you're going to be building glucose. So the fact is you need ATP and NADPH. You need these high energy carrying molecules because actually building glucose, this whole process is endergonic. Not only is it endergonic, it's basically also going to be anabolic. So anabolic, if you forgot, anabolic means to build. So you're actually building up glucose. So let's kind of jump ahead then and revisit. If you look through this Calvin cycle, we're still in a chloroplast, and this is going to be where the glucose is going to be built. So kind of getting this dialed in, let's figure out what we need to know about the Calvin cycle. Okay. So it's sometimes referred to as the light independent reaction. Do not call it if we have the light reaction, don't call this the dark reaction because it can happen at all times of the day. So we're going to just refer to it as the light independent reaction. So Calvin cycle, no other interesting names besides is just talking about the Calvin cycle. Okay. So I am going to talk about this one enzyme. It's one you should know, and that is going to be Rubisco. And that's going to be an enzyme that combines, so an enzyme that combines CO2. And if you look through here, let me just erase this real quick. If you take in CO2, CO2 comes in using the enzyme Rubisco. It's going to combine with this adjacent molecule called ribulose bisphosphate. And that's going to be referred to as phase one, which is going to be carbon fixation. If we're talking about fixation, if you don't know what that word means, fixation, I like to think about it as just building. It's an easy way, right? I'll say building your carbon molecule. So in this, if you look through the phases, there's three that you should know. One being carbon fixation, then we got the reduction, and then you got the regeneration. So in terms of this, let's kind of at least analyze what's happening here. So if you look through this, like I said, carbon fixation, we have reduction, and then we have the regeneration. Now, there's other intermediates. You do not have to know them. I wouldn't even stress on understanding the names. So let's just kind of take a quick second and analyze this. Like I mentioned earlier, Rubisco, it's worth knowing what that is. It's the one enzyme you should know that carbon dioxide combines with this molecule called ribulose bisphosphate, also known as RuBP. And that's going to be the fixation. Without Rubisco, you couldn't actually combine CO2 from the atmosphere, okay? Now, I'm going to basically break this down as your book does, where we have phase one, which is the fixing of carbon. When we say we fix it, like I said, we're building that carbon molecule. Then we're going to actually reduce it, as the name implies. Reducing means you're gaining electrons. So keep track of where these are all coming from. So understand, well, where did ATP come from? Well, it came from the light reaction. Where did NADPH come from? That also came from the light reaction. So basically now you're cashing in all this ATP and NADPH you used to basically provide all this energy. Now notice you spit out, I don't want to say spit out, but when one turn of the Calvin cycle, you produce a 3-carbon molecule. And that's going to be referred to as glyceraldehyde 3-phosphate, but that's not a complete glucose molecule. So hopefully you get the idea that as you do one turn of the Calvin cycle, that's not a complete glucose molecule. You actually need 2 turns of the Calvin cycle. So let me show you this animation first, okay? So it's nice to kind of introduce this and take it slow. There's going to be a lot of moving parts. So this is just reminding you ATP and NADPH was made in a light reaction. Okay, we're going to use this to actually power your Calvin cycle. Now, basically tells you over here that the Calvin cycle begins with carbon fixation. Okay, again, like I mentioned, you're basically going to be building this carbon molecule. Now notice 3 carbon dioxides at a time come in. It's going to combine with 3 ribulose bisphosphates. This is using that enzyme Ribisco, and then you build these 6 3-carbon PGA molecules. Okay, so basically, if you didn't catch that, 3 CO2s came in, combined with 3 ribulose bisphosphate, and then basically you actually built 6 3-carbon molecules. That's your carbon fixation. So in terms of that, let's move on to the next part. From the light reaction, we're going to use to actually add energy here. So basically, this is going to be helping provide energy to build this. Okay, so it tells you the first high energy phosphate groups are added. We got to click on the molecule that provides them. So we have to add in phosphates. So of these, we got a choice. I'm going to select ATP because that can actually donate the phosphates. So I'm basically adding more energy to this. I'm phosphorylating it. Okay. Then it says that the electrons are added in the next step. So of these, who's going to add the electrons? Well, that's going to be your NADPH. Okay. So using NADPH, notice it's adding electrons. I'm adding more energy to these intermediates. Now, this is when I actually reshuffle and make glyceraldehyde 3-phosphate, G3P. Okay. Now, keep track of what's going on here. So I haven't lost anything. All I've been doing is I fixed carbon, meaning I built it up. I then added energy. I added ATP and NADPH. And now I have 6 of these 3-carbon molecules. Now watch what happens next. One of those G3P molecules leaves, okay? It's only 3 carbons in length, okay? Now what's gonna happen to these remnants, I'm gonna reshuffle them using some ATP and remake that ribulose bisphosphate, okay? For more CO2 to come in. Now notice, I didn't make a complete molecule of glucose. So jumping back to this, let's just kind of talk about what we saw. CO2 came in using Ribisco. I made 6 of those 3-carbon intermediates. Okay, that's my carbon fixation. I added ATP and NADPH from the light reaction, right? And then on this last step, after I've spit out 1 G3P, I add some ATP and remake ribulose bisphosphate. Why do I have to redo this? Well, I need more CO2 to come in. Now, if you look at the numbers here, 3 carbon dioxides get the Calvin cycle started, okay? 3 at a time, combines with 3 ribulose bisphosphate. Now, rather than make 3 6-carbon intermediates, it gets quickly reshuffled into 6 3-carbon molecules. So that's where this 6 is coming from. So don't let that freak you out. The 3 in front here is talking about the name. The name of this molecule is 3-phosphoglycerate. So keep track of the smaller numbers. 3 CO2s combined with 3 ribulose bisphosphate actually makes 6 of these 3 phosphoglycerates. So don't let that freak you out. Now, in terms of that, when you get to that regeneration phase, the whole point of this is you have to regenerate ribulose bisphosphate, RUBP, okay, to reshuffle it. So, like I mentioned earlier, 1 turn of the Calvin cycle doesn't make a complete molecule of glucose. So let's take a quick look here. To do this correctly, we have to add in another round of the Calvin cycle. So 3 more carbon dioxide molecules come in. I'm going to fixate it here, right? This is your carbon fixation. Now I'm going to reduce it. This is the reduction phase. I add ATP. I'm going to add some NADPH. I'm going to lose 1 more, and this is really, really great. I'm going to lose 1 more of those G3Ps. Now I regenerate my ribulose bisphosphate, and check out what's going to go on here. Notice that that quickly converted into glucose. So I took all of those 6 carbons, and I actually now have a complete molecule of glucose. Okay, so that's the building of the sugar. So really common things you want to be aware of is, where did ATP come from? Where did NADPH come from? And where did your glucose come from, right? So let's jump back to this. Really common test questions that I see a lot are going to be things like, okay, if we followed, like if we radioactively trace the carbon from the carbon dioxide, that carbon from the carbon dioxide is going to end up on the carbon in your sugar. So they've actually done experiments where they can actually trace that carbon. So that carbon from CO2 can actually end up on the sugar. Okay, like I mentioned in the previous video, what is the point? Why do plants have to make glucose? It's not just to feed us, as a lot of people would say. The plants need to make glucose themselves because they need to make glucose for doing cell respiration when they grow in the roots. They also need to make those carbohydrates for their cell wall as cellulose. So that's the Calvin cycle. So it's kind of showing you over here that this is taking place in the thylakoid. This whole thing is a thylakoid. The fluid outside is going to be called the stroma. So it takes place in the stroma. Okay, thylakoid, the fluid inside of the thylakoid is called the lumen. The fluid inside of the whole chloroplast is going to be called the stroma. So don't lose track of that. And that's what it's showing you here. That's the location. Okay. Now, just to kind of revisit this, I want to take a second and make sure you guys see this. Notice that I only need 6 NADPHs in the reduction phase. But when I look here, I need 6 ATPs. So this is my fixation. In the reduction phase, it's a 1-to-1 ratio. But if you look over here, I actually have to use 3 more ATPs. So this is important for the next thing I have to briefly mention is that more ATP is needed than NADPH, right? So for one turn of the Calvin cycle, I actually need 9 ATPs, only 6 NADPHs. So that's where this comes into play, which is going to be called cyclic electron flow. So I'm going to jump back to the light reaction, right? So remember in the last video, we talked about these electrons jump from one protein to the next, they get excited, and the electrons end up on NADPH. Well, notice that you can actually you do make some ATP in the light reaction, right? And the bulk of that ATP, well, all of that ATP goes to the Calvin cycle. Now, we get into this thing called linear versus cyclic electron flow, okay? Now, basically what's gonna happen here, like I just mentioned a second ago, is sometimes the cell, or I should say the chloroplast, has to make more ATP than NADPH, okay? Now to do that, it basically shuts off that first photosystem, which is called photosystem 2, and it's only using photosystem 1. So rather than having the electrons go to NADP plus, it's not going to. The electrons get shuffled into these other cytochrome complex, and it just shuts this off, and basically the electrons are used to pull in protons and make more ATP, okay? Now, if that made no sense to you, at least understand this. What is the point of the cyclic AMP, or what is the point of cyclic electron flow, I should say? It just produces ATP. You're only using photosystem 1, and it's used to meet the ATP demands of the Calvin cycle, okay? So understanding that is significant to understand. So taking a quick second on this one, that is the light reaction. Let me show you and end this video off with the bio film on this. And feel free to go back and replay my explanation here. But if you look through this whole process, it's not too terrible. That was the light reaction. Let's take a quick look at the Calvin cycle and let's see what's going on. So this is showing you glucose that has already been produced, so don't fixate a lot on those. But when we think about how glucose is made, there's your CO2, okay? This is talking about your ribulose bisphosphate. So this is basically going to be that carbon fixation, adding electrons, adding electrons, adding electrons, adding electrons, okay? So I've actually added some energy. I've added some ATP as well. This is where I'm actually going to be essentially losing some of that G3P, right? So that's not a complete one. So this is just showing you the one G3P. It's going to combine with another G3P, making a complete glucose molecule, and it looks like you're building this nice polysaccharide right here. So that video is not as informative, in my opinion, as this one was. So let me just kind of revisit this to end this whole video. Remember, CO2 comes in. This is your carbon fixation phase. No energy is needed there, but it did take that enzyme Ribisco. This is where you add ATP. You're adding NADPH, right? You're going to lose one of those G3Ps. As that leaves, you're going to basically regenerate that ribulose bisphosphate, RUBP, and it takes ATP to do that, okay? And this lets it go for another round, all right? So that is the Calvin cycle of building glucose. We'll see you in the next video.